Well, hey everybody, welcome to chapter 17, part 2. This might be part 3 or 4, depending on the order in which you watch them. But welcome to my living room and host. We are going to walk through specifically 17.3, the cracking of the code. So we got a quick skirt through all of Katrina's slides real quick. 17.3, cracking the code. So what code are we even talking about? So cracking the code really talks about the genetic code. And just a reminder that the genetic code is the set of rules used by living cells to translate information encoded with genetic material into proteins. Most of us know this genetic code, this amino acid sequence chart right here. So there's two different kinds. There's the kind that I know how to use, which is more the box thing. I found a lot of circle versions while I was looking up different images. So these are both from one of the genome interpretation sites. And these are our RNA codon tables. So how did we get these little charts? Because you and I only know these little charts. But somebody had to define them. So how did they do that? So there was kind of 3-ish major steps. I say 3-ish because number 3 has more than one step kind of included. So these steps led to cracking the genetic code. But within these steps, there are hundreds, maybe thousands, or even hundreds of thousands of experiments that went into cracking this code. So this is more of like an overview slide that we will be working throughout my explanation of what all happened in 17.3. So there are 3 steps: the Mol Polymer step, codon composition, and then codon sequences. So there are overall a lot of words on this slide that we aren't going to talk about quite yet because I kind of want you guys to use this as a review slide. So at the end of this little dealio, when you need a refresher on 17.3 or you need like a quick reference guide, this is going to be your deal right here. It's nice and color coordinated, so hopefully you like it. So the first thing we're starting with is how random synthetic RNA polymers direct protein synthesis, aka 17.3 part 1. So in this step, this is step 1. So Nuremberg and Mathai, sorry Jensen if that's wrong, worked with the synthesis of RNA templates and the corresponding protein polymers they created. Their experiment was incredibly important. It was really the first of its kind that led to this cracking of the genetic code. So we're going to walk through this experiment step by step. So first, let me move myself again. They prepared what's called the translation extract. So they were produced from E. coli and had no endogenous mRNA. That endogenous mRNA had to be removed in order for the cell's translation machinery to work with all the RNA templates they were adding, because they wanted to add their own template. So this was done by using RNase to destroy all of the pre-existing mRNAs that were in the cell, because your cells all have mRNA. And then they used DNase to prevent more mRNA being created by this native cell. They were using E. coli because I feel like we've all learned at this point in life that E. coli is just the secret to science. And so once this was done, they used the enzyme polynucleotide phosphorylase because it can synthesize RNA. But a really important thing about this is it doesn't need a pre-existing template. So what does that really mean? That whatever you add to it, it will do. It will make it in lab conditions. So this translation extract that I was talking about, which if you can't see is, boom, nailed, had color coordinated amino acids being added. So then the ones they added were they started with just poly-U, which means they only added little U's to the mix. Protein synthesis then transpired before they let it happen. They added a certain acid. The book just call it an acid, which is really general, but specifically this acid created a precipitate in the bottom of the tube. This precipitate was then collected and tested for the presence of the radioactive amino acids they added, which have been turned into long polypeptides. They found that all the U's, so a U codon, produce only phenylalanine, which means that UUU would always produce phenylalanine. They then did this again with only A's, finding lysine, then C's for protein, and G's for glycine. So that was really big that they started and found the first 4 identified codons. Now, we have some codons, but as you and I know, we have a little bit more than 4 codons, just in general. So our next question is, what comes next? We started with the homopolymers. Now we're going to look at copolymers. So they needed to kind of go up a step. So they use the same polynucleotide phosphorylase, which was done by Severe Ochoa's group. They use copolymers. It's the same setup. They use the same enzymes. They incorporated NDPs into RNAs randomly. To manage this randomization, they started with just A's and C's, so kind of took 2. They used ADP and CDP in a 5-to-1 molar mass ratio. So that ratio is really important because this allowed for this RNA to be produced 5 more times with A than with C. So they deduced the following distribution, which, let me click again, right there. You'll see that you have A subscript C in their results because this is due to the fact that polynucleotide phosphorylase polymerizes NDP randomly. You cannot assign codon sequences, only codon composition. So if you think back to that original slide, which had the 3 steps, this is step 2, the codon compositions. We're starting to see mixing of letters, those A's, C's, U's, and G's, but we're not really sure. But what did they do with this information? Because it's still really valuable. They were still able to learn a lot. They compared what they observed versus the expected frequency as from the relative amounts of amino acids in this precipitated protein product. So they're still using that same add an acid, get a product, test the product. They could deduce that the respective codon compositions specified certain amino acids. They found that these combinations of A's and C's produce a synthesis of polypeptides containing asparagine, glutamine, histidine, lysine, proline, and threonine. So this concludes our step 2, as the book quickly discusses how various RNA copolymers experiments led to the discovery of the composition of about 40 codons. So we definitely are doing more than just the OG 4 we found, but we're still only at 40. So this still really leaves us with the question of how do we actually define sequence? So, where's my mouse? There we go. RNA polymers of defined sequence complete the code. So this is really important because chemical synthesis of short nucleic acids of a sequence was needed to complete this genetic code. This is our 3rd step. This is kind of where we get into the 3-ish part. This is like 3rd step, part 1. So Nuremberg was back at it as he discovered a way to identify an amino acid associated with a short synthetic codon. So he's still kind of using the same first experimental setup, but there was one major change. He found during protein synthesis, the nascent protein, aka this protein being created, stayed attached to the ribosome. But it fell off when he started adding the acid we talked about that forms the precipitate, which is from that original slide. This acid created the precipitate he tested, but he found that if he didn't add the acid, instead use a filtration method that worked with a nitrocellulose filter, it would bind the protein, but not RNA. So that ribosome would bind. They were able to find out about trinucleotides, and they could use them in this setting, which are super important because at like a parallel pace, Maxine Singer found a way to develop synthesized RNAs in a defined sequence. So not only were they able to start looking more at the RNA, they could also decide what the RNA would say. So now with the ability to create those trinucleotide sequences, they could begin testing specific sequences. So they were able to use individual reaction mixtures and pass them through this filter, trapping the C amino acyl tRNA bound to the triplet RNA codon. So this part B of this figure down here, which let me grab my pointer, right there. summarizes how they worked with U's, A's, and C's. And then, so this, like machinery, this experiment has taken up, and similar studies were done and found approximately 50 codons for specific amino acids, but still could not bind some of them because they weren't bind tightly, so they couldn't really work with this filter. So there's still more information that we're missing. We have quite a few codons. We're starting to get at codon sequences, but there's still just a teeny bit more, which is why I said 3-ish steps. Our third step in cracking the genetic code has a little bit extra. It's like a 3.5 step. So the codon sequences, take 2. So H. Gobine Corona, sounds a lot like Corona, fun fact, came into the picture. He helped crack the code by developing ways to synthesize short polyribonucleotide sequences with a defined sequence of 2, 3, or 4 nucleotide repeats. So that repeat part is super important. So he would then amplify those sequences to produce long repeated sequence polymers. But how did this help? Why do we really care? He solved our subscript problem that I was talking about when it was like A2C or C2A or mixing all the other letters in there too. Because of the polypeptides he synthesized on this RNA copolymer, it contained equal amounts of threonine and histidine. So we're talking specifically the A's and C's. So in combination with a couple other studies, they could figure out that CAC must be histidine and ACA for threonine. Through all this work, he was able to identify 61 of 64 possible codons, and also, in this just a little side project, confirm that mRNA is also read 5' to 3' direction using the same repeating sequences. So he also used di, tri, and tetranucleotide sequences. So if you look at this chart down here, let me put my pointer back on, he was able to find different copolymers, but also homopolymers and repeating copolymers. So this was key to figuring out codon sequences. So then, what kind of happened after that? So we have quite a few sequences, but the story's not over. But we kind of completed our 3.5 steps into cracking the genetic code. But there's been some shifting over the years. And what is that shifting? It's a mutation. So there have been mutant proteins that have resulted from a single nucleotide substitution. In all of these cases, the incorrect amino acid in the mutant relative to the wild type or the quote-unquote normal type could be accounted for by a single change in a single nucleotide based on the genetic code. So one example the book talks about, which I thought was really cool, was sickle cell anemia. I also think that's kind of cool because we talk about it in quite a few science classes. Like I know we talk about it in cell bio. Pretty sure we talked about it in a different bio. We talk about it in bio 120, probably bio 130. So sickle cell anemia happens when there is a single amino acid change causing glutamate to be subbed for valine. So just an AGU becomes a UGU when it's transcribed in RNA. This change in the amino acid sequence, as you can see on the bottom of my little picture dude, causes hemoglobin molecules to crystallize when oxygen levels in the blood are low. So as a result, red blood cells become sickled and get stuck in small blood vessels. That is why, fun fact, the NCAA makes all athletes either be tested for sickle cell anemia or sign a waiver that says because this can become a really big problem when you're working out, which is a situation where you're low on oxygen. And then I wanted to just include one of the resources I use. This is a really, really good recap video. I'm not going to show it because it's like 7 or 8 minutes long. But between this and looking at this, the lighting switches when I click the slide, sorry. Looking at this little deal I made you guys as like a recap, I really think you guys can understand how we cracked the genetic code to get all of these RNA codon things. These sequences show up in like every science class you take, and so it's really important to know how we got them. Good luck, guys. I hope you're all staying inside and are staying safe. So, adios.